Today, our topic is nucleic acid hybridization, blotting technique in screening for recombinants in principle and process of biotechnology, Unit 8, Chapter 4 in botany. So, nucleic acid hybridization, blotting technique. So, the blotting means staining. Staining is the meaning. So, here they used an analytical tool as this blotting paper, that is blotting technique. Why they are using this blotting technique? To find out the desired DNA or RNA fragments from a group of number of molecules. So, they had given some group of items. In that you are going to find a particular trait. So that trait was seen in a particular gene. So that gene has DNA or RNA fragments. So for this they are using this blotting technique. So blotting refers to the process of immobilization of samples, nucleic acids or solid support, nitrocellulose or nylon membrane. So here in blotting technique, that is blotting is a process to immobilize the sample which may be nucleic acid, and this immobilization, that is movement is not seen, movement should be restricted in the sample, and this can be done in a solid support which you can use the nylon membrane or nitrocellulose. So the blotted nucleic acids are then used as target in the hybridization experiment for the specific detection. So why this blotted nucleic acid? So what this purpose we are doing? So we are going to find out the desired gene from the hybridization techniques. Okay, that is our target. Types of blotting techniques: southern blotting, northern blotting, western blotting. Keep it in mind. Southern blotting is used to transfer the DNA to nitrocellulose membrane, and northern blotting is used to transfer the RNA to the membrane. Western blotting is used to transfer the protein to nitrocellulose membrane. Okay. So the agriculture diagnosis. So this is to treat whether the crops is infected by the pathogens. So to find out that, there are two methods, ELISA and DNA probe. So the ELISA is the famous test which is acting as a confirmatory test for the HIV AIDS. ELISA means enzyme linked immune sorbent assay. So this is used to diagnose the pathogenic species using the antibiotic. They are doing this, okay, and the diagnostic agent. So they are used to find out the pathogen using an antibiotic, that is antibodies, and the diagnostic agents. So ELISA can easily find out the virus and other pathogens in large scale also. So DNA probes, if you come across the word probe, means you can blindly write it, that is it is the radioactive isotope is used here. Okay, so that means the northern and southern blotting is used, that is to identify the viruses and other pathogens. Okay, so the two test methods are used for to control the pathogens which causes a great loss in agriculture. So for this they are using the diagnosing the disease, ELISA and DNA probes. Southern blotting technique, DNA. As we talk about the types of blotting, the southern blotting technique, it is used to transfer the DNA from agarose gel to nitrocellulose membrane, and this was introduced by Southern in 1975. So the name of this technique came after the Southern blotting technique. So steps involved in Southern blotting technique. First, the transfer of DNA, that is from where to where, agarose to nitrocellulose filter paper, and this was taken place by capillary action, by a drop by drop, using a capillary tube, a minute tube. So you know the capillaries are the blood vessels, that is they are using for the blood vessels, that is in our body also the capillaries are, so it is a very minute than arteries. So like that capillary action, the transfer is taking place. And a buffer, so that is a buffer is used, sodium saline citrate. Yes, see this they may ask in your 1 mark. Sodium saline citrate is used as a buffer, so in which DNA is highly soluble in it. So the DNA is soluble in this buffer. SSE, sodium saline citrate, they may ask in your 1 mark as well as in your NEET exam. So be drawn up, it can be drawn up through the gel into the nitrocellulose membrane. So this buffer is soluble, this buffer is used to soluble the DNA, and this can be drawn out, that is the drawn out through the gel while transferring to the from the gel to the nitrocellulose membrane. So by this process, the single stranded DNA becomes trapped in the membrane matrix. So in the membrane matrix which was caught, the single stranded DNA was caught. So this DNA hybridization with nucleic acid can be detected by autoradiography. So that is the transfer was taken place and it was the desired gene has to be detected with the help of autoradiography. So autoradiography means it is a technique to capture the image formed in a photographic emulsion film, that is emulsion, while passing the light on it, emitting the light on it. So you can see the labeled radioactive which was placed on the unexposed film. So that procedure is called autoradioactivity, and this is the blotting technique apparatus. This is the support block, and this is the filter paper, and it contains the buffer. Here also that is the transfer buffer is collected, and this is the gel plate. So the gel plate, agarose plate, on that they are placing the nitrocellulose membrane, and then filter paper. Again you can see the paper tissues which was placed on the paper tissues. They are plating the glass plate and weight for transferring the material, that is the DNA from gel to filter paper, that is nitrocellulose filter paper. For this they are using the weight. Okay, and now we can see the Southern blotting steps, how it works. Okay, the first step is the DNA is cleaved by restriction endonucleases and transferred to an agarose gel. The fragments are separated by gel electrophoresis. So the desired gene has to be cut with the help of restriction endonucleases. So after that restriction endonucleases cut the enzyme, that is that enzyme cuts the DNA, and it was transferred to this gel, and it was transferred to this the gel that is the gel. This was transferred to the gel electrophoresis instrument. So the second step, the gel is soaked in a alkali solution to denature the double stranded DNA and then placed on a flat platform in a dish containing buffer. So here you are adding a gel, that is in gel you are adding an alkali. Why you are adding an alkali? To remove the double standard, that is you are denaturizing, that is the normal natural model is double standard DNA. So that double standard DNA you are going to make into a single standard. So you are denatures the DNA using the buffer. And the third step, the membrane is positioned on the top of the gel. So this is a membrane is placed on the gel. So nitrocellulose, you can see this nitrocellulose membrane or other membrane, gel and blotting paper and the alkali solutions are kept here. So this is the exact transfer method using the Southern blot. Okay. A buffer drawn up into the top layer of the blotting paper passes through the gel, carrying the DNA onto the membrane. So the buffer is passes up to the top of the gel, and which can carry the DNA to the blotting paper. That is, the blotting paper is nothing the nitrocellulose membrane. And next step, so on the next step, in the next step you can see, okay, this is the next step. So the next step, the DNA on the membrane is fixed. So you can see the membrane was fixed. This is the membrane and this is the gel, and you can see the bands on the membrane. So now the DNA is fixed on the membrane. And sixth, the next position, placed in the hybridization bottle with the solution that contains an radioactive labeled probe and gently rotated. So it was taken into the radioactive probe, okay, in the bottle. So to find out the DNA, desired DNA. So there is a gentle rotation was taken place after the DNA was inserted in, that is taken in this bottle. A gentle rotation was taken place, and in this bottle you have a radioactive probe also. The probe binds to the complementary DNA fragments on the membrane. So the probe is a DNA fragment, and the whatever we take inside this bottle is a single stranded DNA. So now the probe and the DNA will mix us and it forms the complementary strand of the DNA. Okay. So the size and standard. So this is the next step, that is the filter paper contains the DNA bands, and while taking the autoradiography, that is the photographic film, you can see the size and the standards this side. So with the help of that you can identify, okay. So with the help of that we can find out the agarose gel electrophoresis is transferred, that is the DNA is transferred to the filter membrane through these strips. And now you can see the last step, that is autoradiography detects the fragments with the probe attached. So here the probe is attached and you can easily identify it. So these are the steps involved in Southern blotting technique. You can draw this diagram and you can write as it is the 6-8 points. You will surely get 5 marks. This they may ask in 5-mark question also. Next, the western blot. As we talk about northern blotting, as we talk about the types of blotting, the northern blotting, transfer of RNA into the membrane. So here we won't use nitrocellulose membrane. So we will use another membrane for it, what we will see. So this was given by Alvin et al. In 1979. So in this procedure, the RNA doesn't binds with the that is filter paper from the transferring the agarose gel to nitrocellulose filter paper. So the transfer of RNA from gel need a special or specific filter paper for the transferring the northern blot hybridization. The filter paper used for northern blot is amino benzyloxy methyl paper. So this they may ask in your NEET and in your board exam. So amino benzyl oxymethyl paper, so which can be prepared from the Wortmann 540 paper. So the amino benzyl oxymethyl paper is prepared from Wortmann 540 paper, and this paper is used for the transfer of RNA from agarose gel to the filter paper. The filter paper used here is amino benzyl oxymethyl paper, which is prepared from Wortmann 540. This they may ask in your NEET exam as well as in your board exam in 1 mark. Next we move on to the western blot. So the western blot, transfer of proteins to the nitrocellulose membrane. So the protein can be identified using the probing. Probing means the radioactive one. Here they are using the antibody to find out the specific protein to which the antibody is prepared. So the antibody probe is used to find out the specific protein for which the antibody is prepared. So that is called western blotting technique. So there are differences between the techniques used. Southern, northern and western blot. Name. The southern name of the inventor. Northern is a misnomer, that is opposite to southern, northern. So it is a misnomer. And western is a misnomer. It is also a western. So northern, there should be northern part, southern part and western part and eastern part. So like that they had given this name. Separation. Southern blotting is for DNA, northern blotting is for RNA and western blotting is for proteins. Denaturation needed in southern blotting but northern blotting not needed because it is already single stranded, and in western blotting the proteins, so denaturization has to be needed. So the membrane used in southern blotting is nitrocellulose membrane or nylon membrane, and in northern blotting they use, that is aminobenzyloxymethyl, and in western blot nitrocellulose membrane. Hybridization. In southern blotting, DNA hybridization. In northern blotting, RNA-DNA hybridization. In western blotting, protein-antibody hybridization. Visualization through autoradiography in southern blotting. In northern blotting is also autoradiography. In western blotting, through darkroom. So if they ask the differences, you can write this. Southern, northern and western. Even if they ask any, it can be act as an key points to easy to remember it. So if you have no time while writing the examination, the time is going to over. You just, if they ask the southern blotting, you just give these points also, you will surely get marks. Okay. So hope you understood. Thank you for watching the video. Give a thumbs up, share and subscribe to Science Easy Tech channel.